Cell organelles. We'll look at function as well as structure. And obviously they go hand in hand because obviously structure determines function, right? So what we have here is the cell membrane. Notice how it has a double phospholipid bilayer. So it has one layer over there, one layer over there. What you are looking at there is the head. Okay, so these heads, heads are hydrophilic. All right, hydrophilic. Why? Because philic means not afraid of, and hydro means water, right? So it's not afraid of the water. So the environment on the outside there, here, all around it, it is the external environment, and it has a high concentration of water, okay? And it's also considered aqueous as well because certain things are dissolved within it, whether it's ions or different covalent molecules, okay? It still has a high concentration of water, and because of that, because these heads are always maintaining contact within that environment, it is not scared of it, so it's considered hydrophilic, okay? It actually likes the water. What we have on the inside here are the tails, and we consider these as hydrophobic because, of course, phobia means scared of, so it's scared of the water, the hydro. Okay, so that's why it's staying within the inside, so it stays away from this environment of aqueous, right, or of the water. So the tails are actually very friendly when it comes to certain molecules that are fat substances. So let's say certain lipoproteins, waxes, cholesterol, okay, all of these things, um, or molecules that are maybe of non-polar specificity, they can actually be able to get through, and they're able to get through there a little bit easier compared to other molecules. Again, provided that their size isn't too large. But again, we're going to learn more about this a little bit later on. But other things that are polar and like to be dissolved in water can also go through as well, because obviously the heads up there are hydrophilic. Okay, so there is nuance to it, but again, we'll look at that a little bit later on in the next unit. What I really want to get into the specifics here is going to be in this area with the functions. Okay, so like I said, with the heads and the tails, they allow certain materials to go in. It also has some embedded proteins. Embedded proteins, okay? So there's some embedded proteins here, etc. And some of these embedded proteins have very specific functions, okay? Some of them act as receptors, for example, like smell or taste, right? So like odor receptors, for example. Communication messaging, transportation of materials, or a gatekeeper for certain ions, right? So like I said before about the size, right? If an ion size is particularly very large, it can't actually go through this barrier very easily. It actually needs some support, okay? Whether that's through energy, right? Through the breakup of ATP, which we'll be looking at later on, or maybe through facilitated diffusion. Again, there's different ways because sometimes it'll be easier than others depending on their size. Okay, so I'll just put here some various functions, okay? Various functions. And it actually acts as protection, okay? So I'll just say protective. Okay, it acts as a protective layer to make sure that not every single molecule can go through, right? That can be damaging or harmful, right? Again, some substances could be very harmful, like LDL cholesterol, for example. Let's say low density, or even worse, VLDLs, right? It's a very low density lipoprotein. These lipoproteins destabilize the cell's membrane. Whereas good ones, or like HDL, like high density lipoproteins, they're a type of cholesterol, type of fat, okay, within the cell that helps stabilize it and helps bring certain fats towards the liver to be broken down. So again, this one would be the healthy version, which you want a lot of. What you don't want are low density or very low density lipoproteins. They actually increase APBO levels, APOB markers, okay, and these levels are causative when it comes to increase in serum cholesterol, which is a predictor of atherosclerosis. Again, what am I trying to say? What's the point? The point is they're very, very selective to make sure certain things can go within these barriers at certain times when needed, and it absolutely blocks out certain things that we absolutely do not want. Okay, so it's a selectively permeable. It selectively allows certain things to go through and certain things to get out, but it absolutely does not allow things that are harmful to be able to go through. So maybe I'll write this over here in a star here. It's selectively permeable. All right, and what is this image? I know this kind of looks like a biopsy of the whole cell, but that's actually not what I'm trying to go for here. I just want to show you where the cytoplasm is in reference to the actual cell, okay? Because if none of these were there, it would just look pretty much empty, right? But that's what I want you to focus and and hone in on, right? So this area over here that is not the dots and is not the organelles floating around, okay? All this empty space around here that is holding everything together, kind of like a jelly, right? Like a pudding, okay? So all the stuff around here, right? All of these are the cytoplasm. The cytoplasm gel is very, very important, okay? So it's a jelly-like substance. I'll just put here, it's jelly-like. Okay, and it fills most of the inside of the cell, okay, and it holds organelles together, okay? It's mostly water. It has some proteins, okay? So some proteins, ions, etc., that are dissolved within the water. And what did I say in terms of the function, right? I said that it holds things together, right? Holds things together to make sure that they don't go off in a direction that is farther away, making it less efficient to do reactions. Cytoplasm is actually a very important area of space because protein synthesis actually occurs in this area, okay, on the outside, okay. Obviously this here is the nucleus, the control center of the cell, which we'll be looking at shortly, but pretty much on the outside over here, protein synthesis occurs, okay. So DNA replication, it actually occurs on the inside here. In order for it to get out, right, the particular code that gets transcribed, the mRNA needs to leave, and when it does leave, the cytoplasm, which we're looking at over here, is the site where protein synthesis occurs. And obviously you are made out of proteins. It's the structural building blocks of you. Okay, it's also important to know that it's a transport medium for nutrients, okay? So it's a transport medium for nutrients. It maintains optimal environment, okay? So particularly temperature is a big one, okay? It maintains an optimal temperature. Again, because cytoplasm made mostly out of water. A good thing to kind of think about if you've taken chemistry already is specific heat capacity, right? Because water has a relatively high specific heat capacity, there's not going to be sudden changes in temperature because it's going to be difficult for that material to change temperature abruptly. Okay, because of that specific heat capacity. Okay, so again, it maintains that optimal environment, that optimal homeostatic environment that we want it to be completely or more or less balanced most of the time. We don't want any drastic changes quickly. It is an area that helps with cell metabolism. And lastly, like I said, it provides some suspension for the organelles to be nice and tight and not move around, okay? Now what is this here? This here is the nucleus. I'm sure you've heard of the nucleus before. It's the most important organelle because it houses the DNA, which is the hereditary information. It pretty much controls who you are and what you're going to become. Obviously, it is a round structure, and it has some pores. Obviously, it allows certain things to go in, certain things to go out. And it has a nuclear envelope around it. Okay, so obviously that there is a nuclear envelope on the outside. Now you can see some distinguishing features within the envelope. You see okay, these are some embedded proteins. So these embedded proteins have those various functions that we talked about earlier, right? About acting as a receptor, communication, allowing certain things to go in and out. But the most important thing that I want to focus in on here is it contains the chromosomes. Within here, it has the chromosomes. Why is that important? Again, the chromosomes are the DNA. DNA is the hereditary material. So let's look at some important functions here. It is the control center. Everything that is done must happen through the command of the nucleus. Like I said, the pores allow for the transport of the materials, but the most important part that I want to really highlight here in the star here is it regulates cell division. The reason that you are here today, starting from one cell to becoming probably more than 100 trillion cells, is because of cell division. One cell duplicating into another, into another, into another, could not happen without the nucleus. And we're going to be talking about the mitotic and the meiotic divisions later on in the unit. But again, all of this must be set forth and put forth because of the nucleus, because the DNA has that code that tells exactly when to divide. And obviously, you're not going to keep dividing. Otherwise, you're just going to keep getting bigger and bigger and bigger, and that's not what happens. Eventually, we stop growing. And it's all timed by a cell clock mechanism. At some point, when you need to stop growing or slow down the growth, this is all controlled by the nucleus. When you get a cut, however, and you need more skin cells to grow back, the nucleus is, again, that control center to make sure that there is division in that particular area that needs to be fixed. So just some food for thought. All right, here we have some vacuoles. All right, here are vacuoles and some vesicles. This here is a vacuole, this here is vesicle. An important thing to note here for the structure is that they are different sizes. Different sizes. Okay, in a plant versus animal cell, depending on the type of organism. So in a plant cell, it's actually very, very large. So I'll just put here large. Okay, particularly the vacuole, it is ginormous. Okay, for the animal cell, it tends to be quite small, the vacuole, because they have two very different functions. Before I get to their functions, though, I just want to put here as a star that they're both fluid-filled sacs. All right, so let's look at their functions. So how are they fluid filled? Well, in plants, they hold lots of water. Okay, so fluid filled in this case would be the water. But for the animal cell, it more so contains the waste products. So vacuoles and vesicles, they both store material, but I really want you to focus in here on what the vesicles particularly do that the vacuoles do not. For the vesicles, please remember that they're the only one that actually transport materials out of the cell or inside the cell. Okay, out of the cell or in the cell. And we're going to be learning more about endocytosis, exocytosis pinocytosis, phagocytosis, all of this stuff, okay, and how and which types of molecules can get in and which ones can get out. But just know that it's the vesicles that are going to be the ones doing that, not the vacuoles. Okay, so because the vesicles transports things. So if this was your cell over here, okay, this whole thing, let's say, was your cell. Okay, so this cell, um, certain things need to get out. Okay, so it's going to go towards this boundary over there of the cell, and it's going to allow certain things to go out. Okay, based on what it has locked up in there. And if something needs to come in, okay, so let's say there is a molecule over here that would be very, very beneficial for the cell to have, okay, these vesicles will be able to take that one and embed it within the cell membrane and for it to come inside into the cell. Again, the vesicles would be responsible for that to happen over there. And because it's spending so much time in and around the border, it makes sense that another function of the vesicle actually is to maintain buoyancy control or buoyant control. Vacuoles actually maintain some turgor pressure as well, but that is outside the scope of this course, so we won't even talk about that. Next here we have the mitochondrion. As you can see that there are some folded layers, okay, on the inside. Why are there so many folded layers? Well, the more folds you have, the more you have in contact with the environment that it needs to have reactions with. So the folded layers, it's kind of like the layers in a small intestine. Okay, in a small intestine, it has villi and microvilli. Again, it also has that folded kind of projection because the more contact it has in that certain particular period of time, the more reactions can happen within that period of time. So it's very, very efficient when it comes to like digestion, metabolism, etc. So for mitochondrion, we'd say that same thing, okay? And it kind of looks like a bean. So I'll say it's kind of like bean shaped, okay? And it's smooth on the outside, obviously, as you can see over here. But because the inside has so many folds, okay, entangled and intertwined, obviously it's not very smooth on the inside. It's actually kind of convoluted, okay, because of those layers. I also want you to know that it has ribosomes. As you can see, those dots, you see those dots over there, lots and lots and lots of dots. Those dots are the ribosomes. It's a organelle as well, which we'll be looking at shortly. And it has some ATP synthesis products. And this is crucial, okay? Let me explain kind of why that's important. For ATP, okay, why is it so important? ATP, it is adenosine triphosphate, and when one of its bonds of its phosphates breaks off, it releases a ton of energy. And that release of energy allows for us to be able to do work, usable work, thinking, exercising. That's why we kind of call, a lot of people call it the powerhouse, because all the things that you need to do to power through, like I said, work out, think, study, to do daily activities, it requires that conversion of chemical energy into that usable energy that we can use to push things and do things. It all stems from ATP. I'll just put over here, allows usable energy. All right, from chemical energy. Now, if you're kind of confused what I mean by chemical energy, so for example, when you think of the word cellular respiration, right, mitochondria helps with that. Okay, so let's say, here, I'll put it up here. It's very important, so I'll put a star here. For cell respiration, there's many different types of cell respiration, but I think the one that you're probably most aware of is the exchange of the gases of oxygen and carbon dioxide, right? So obviously we want oxygen, we want to get rid of carbon dioxide, especially during times of exercise. So when I guess when you focus in on the alveoli that are within the lungs, there is a particular medium that allows the oxygen from the lungs that went through the bronchi, through your throat, right, through the trachea, to diffuse into the blood. That's where the circulatory system and the respiratory system interact, right? You want that oxygen from that external environment to go into the blood, right? And at that same precise location, we want that carbon dioxide to be removed at the same time. So cellular respiration, what is the formula for it? Well, when you eat food, because obviously without food, you don't have the energy to do any of this. So when you have that food from the glucose, and you get that oxygen that was just breathed in, what does it do? Well, it rearranges the bonds over here and creates certain products. Carbon dioxide, the one that you do not want, creates a very, very acidic environment when it converts into carbonic acid and releases hydrogen ions. We don't want that, okay? So carbon dioxide gets released and then we breathe that out in the opposite direction. And of course we have water, okay, like sweating it out, for example. Okay, so cellular respiration. In times of work, study, all these things are high demand. And we have a brain that requires a lot of glucose, pretty much runs off solely off glucose. So all that chemical energy that you're going to be getting in from the food and the oxygen that you're breathing in, through cellular respiration, you're getting rid of these things. Okay? Now, so far, what we've been looking at, all the organelles that we've covered, they all fall within animal cells and plant cells. This, however, this guy that kind of looks like the planet Mercury, right? But obviously it's not. It's considered it's called a lysosome. Lysosome. Okay. This, however, is only in animal cells, and there's going to be some other organelles that have some features where it's only for plants and only for animals. Okay, but for now, this is the first one that you've come into witness with. Okay, it's the lysosome. Okay, it's only in animal cells. So it's a sac, and it's filled with liquids. And enzymes. Now, in terms of the size, like if you're probably wondering like how big this is, well, the lysosome, if I were to put it in the middle over here, it's actually larger than the vesicles. I'll just put it here. It's larger than the vesicle, right? Remember, the vesicle transports of certain molecules out of the cell and into the cell, but the lysosome might actually be a little bit smaller than the nucleus. And maybe it's also important for you to know that it actually contains a lipid bilayer. Okay, lipid bilayer as well. Okay, particularly below the membrane. So let's look at some functions. So when we looked at the enzymes here, why is that so important here? Well, the enzyme digests some things, okay? Well, what does it digest? Well, it particularly digests foreign substances and kills them, because if we definitely don't want those things to be floating around and invading our cells, especially if they're very, very harmful, okay? So it digests those quickly and efficiently. Quickly and efficiently. This should be pretty easy for you to remember, because enzymes, why are they very, very efficient? If you remember from maybe previous grades, enzymes decrease the activation energy required for a reaction to proceed, right, to go into process. So obviously having enzymes, I believe it's enzymatic hydrolysis, that the lysosomes have in regards to its particular enzymes, those help digest and degrade certain materials that are extremely harmful that we do not want. And other organelles will come around and pick them up and discard them. And it's important to know any worn out cells or worn out parts can get discarded too. If it's not acting very optimal, or if it's kind of old or degraded or it's damaged, lysosomes help to clear them out, because we don't want those things to be dividing, right? Next we have the Golgi apparatus. Simply put, it's just a stack of flattened membranes. And as you can see, around it are some vesicles, right? So I'll just put here that vesicles are embedded within it. And why is that the case? Well, it sorts and packages the proteins. So sorts proteins, okay? And packages them away, kind of like a mailman. And where do they get these proteins from? Well, they get them synthesized and produced from the rough endoplasmic reticulum, which we'll be looking at shortly. The vesicles, like I pointed out to, right over here, here, right? They help deliver them away and delivers them away. Okay, again, thanks to the vesicles. A cool thing to note, they actually help form lysosomes. It also transports lipids, okay, so fats. The Golgi apparatus, like the other organelles mentioned previously before the lysosome, it's in both animal and plant cells. Okay, so again, it's in both plant and animal cells. Okay, just remember that. What we have over here is the smooth endoplasmic reticulum. It lacks the ribosomes, so unlike the thing that we just saw, the Golgi apparatus, where it had those dots, okay, it had those ribosomes. The smooth endoplasmic reticulum does not have it, so I'll just say lacks ribosomes. Okay, so it's a network of tubes or tubules that run throughout the cell, okay? Now, what is its function? It just makes fats. This one too is found in both plant and animal cells. This here is the rough endoplasmic reticulum. Again, it's found in both plant and animal cells. This one does have ribosomes. And as you can see, those dots are very, very apparent, right? There. That's pretty much all over the place. And again, it has a network of tubules that run in and around the cell. What's important, this one does not make fats, it actually makes proteins. Produces proteins. Okay, and who do we give this to, right? Who does this RER, right, the rough endoplasmic reticulum, who does it give it to, these proteins? You guessed it, gives them to the Golgi, right, the GA, the Golgi apparatus. Here is the ribosome. Now, you're probably thinking, why isn't it just a dot, right? That's probably what we've been looking at the past, minutes. Well, the ribosome actually has many different constituents, and it can make different types of ribosomes. So this one over here that you see here, okay, that had to have occurred within the nucleus, remember? And through the replication process, it turns it into mRNA, okay? So this thing is the mRNA. And using this particular code, transcribed from the actual real DNA from the nucleus, it's going to be turned into the appropriate protein that we want. Now, what is it resting on? It's resting on another type of ribosome here, okay? The rRNA. Now, ribosome and rRNA are sometimes used in literature, and educators kind of use them interchangeably. I mean, it technically is, okay, rRNA is a ribosome, but they're a little bit disjunct as well. I mean, 60% of it is rRNA, the other 40% of ribosome creates other types of RNA molecules. But I guess for the scope of this course, you can kind of think of them as synonymous, okay? So this rRNA here is this ribosome piece, okay? That kind of holds this mRNA in place, the code that we want to be processed into an actual protein. Okay, and what comes in towards the mRNA, it's this substance over there. This is called the tRNA. And depending on the code that is represented on this mRNA in triplets, okay, so that means in every three. So for every three amino acids, we call that a codon. Again, we're going to be learning more about this a little bit later on. I just kind of want to give you a heads up here. So for every codon, so for every three letters or every three amino acids, that codon is going to be read by this tRNA. And the tRNA has its own constituents of code, okay, on the outside. So for every three, so it has 1, 2, 3, 1, 2, 3, 1, 2, 3, okay? As it's reading this mRNA that is being processed, it delivers these codons when it is needed, when it's appropriate, depending on the stage that the mRNA brings it in. The tRNA then matches that. Eventually, they all link up and join together, form a polypeptide, gets released, and then starts folding, and then actually makes a protein, flies away, and then now you have your protein. Okay? So just know that there are many, many moving parts within the ribosome. So I guess I'll just put in stars here. It's made of RNA, okay? Made of RNA. Oh, it's also attached on the rough endoplasmic reticulum, okay, the RER, that is just free floating in the cytoplasm. Let's get into the functions, right? So like I said, makes proteins or synthesizes proteins. Again, from that mRNA. Using that mRNA helps make that protein. This one, again, isn't both. It's both in plants and animals. Guess what? The ribosome is actually in archaea and bacteria as well, which is in a completely different domain than plants and animals. Here we have a cytoskeleton. It is a network of mesh, okay? Mesh of proteins. It's to act as the backbone of the cell. So it gives it structure, it gives it shape, okay? I'll just put structure and shape, SNS, okay? Structure and shape. Helps move the cell itself as well. So of course, you're probably thinking within the cell, what's the most important thing when it comes to movement? Obviously, it's the division, okay? So it supports the cell during that division process of mitosis. Okay, so aids division. Again, also in both, okay, plant and animal cells. So this here is the cell wall. This is only in plants. Please remember that, only in plants. Animals do not have a cell wall. They have a cell membrane, but not an extra layer of a cell wall. Plants have a cell membrane and that extra layer of a cell wall, which is why when you're chewing down on celery or other fruits and vegetables, you can obviously tell it has that extra layer that makes them a little bit more difficult to break down. So it's a rigid layer, okay, of cellulose, also known as fiber. Okay, so I'll just put here cellulose. Okay, it's also semi-permeable. Okay, so what are its functions? Well, it strengthens, and it of course protects. And it makes sure that the cell remains static and doesn't fall over, bend over, or rip off. Okay, it provides structure and support. Okay, so I'll just put again SNS, structure and support as well. This thing here is called a chloroplast. This is also in plants only, and it has something called chlorophyll. Right, so it's a pigment actually found in plants. It has a stack of thylakoids. So these individual circles there are grana. You put them together, it's called a thylakoid. It's very, very important when we get to photosynthesis, because obviously without chlorophyll, within that chloroplast, photosynthesis would not be able to occur. Particularly when we get to the light-dependent phase, the thylakoids are very, very important. It has a double outer membrane. And it's flat discs or oval, okay? Like I said, this is where photosynthesis occurs. In case you remember the formula, it's pretty much the opposite of what we looked at with cellular respiration. We have water, I'll just abbreviate it here. So we have water and CO2. And obviously we need sun, right? So of course there needs to be some sun here as well, sunlight, to create that oxygen. Oops, oxygen. And C6H12O6. That's the glucose, right? Because obviously plants make the sugar. That's why they taste good. And they release that oxygen, which we need in order to survive, in order to breathe in. Okay, so it produces energy through this process. Next we have centrioles. They are barrel-shaped. And it's pretty much a ring of microtubules. It's part of that cytoskeleton that we looked at earlier, right? It was one of those components. And it's arranged perpendicular to one another. I know it doesn't kind of look like it over here within the image, okay? But they are actually perpendicular. Within the inside over here, you're going to be seeing them in a perpendicular direction, okay? To obviously have increased stability and rigidity. So let's look at some of the functions. It's important in mitosis, right? Mitosis, the division of the cell, very important. And it helps produce something called the spindle fibers. Makes spindle fibers. Okay, and what does that do? It splits the chromosome during that mitosis. Important to note that the centrioles are only in the animal cells. Now this one here is cilia. This one here is flagella. They are also both found only in animals. I mean, this kind of gets to a gray area here. I'll give a little side note. It's very important, actually, after I finish off with these two, and a small little note that you would need to remember. They're kind of tiny hair-like projections. I'm referring to the cilia over here, right? They kind of look like a hair and extend from the plasma membrane or cell membrane. It means the same thing. Alright, so these particularly more belong with the cilia, okay? And then this one over here I'll indicate for flagella. It contains microtubules. Generally, both are actually for movement. So if I were to put a big section over here, it's for movement. Some obviously examples, obviously for the flagella, the first thing that comes to your mind, okay, is obviously the sperm, okay? So that sperm has the flagella so that it can help move around. And other or organisms out there, another one could be like Euglena. Okay, it's a type of protist. They need to be able to move around, or certain types of fungi like spores, they actually have flagella as well in order to be moving around. Bacterial cells also have flagella in order to be moving around. Okay, they don't have feet that walk around, right, like animals do. So flagella just helps with movement. Cilia, you wouldn't think it would help with movement, okay, at first glance, but you have cilia in your nose or within your trachea to ensure that if there's any foreign particles that are going through your nose, right, through your nasal cavity that we do not want, that are harmful, okay, they get trapped within the cilia, mucus builds up to help push that away. You sneeze and it gets that gets that out, right? So the cilia actually helps with that movement and pushes it in the direction out towards the outside of your nose so that we can eventually get it expelled. Okay, same thing with the throat as well. If there's any harmful pollutants in the air that we do not want to go within our lungs, the cilia helps push that array of particles with the help of mucus, pushes it uh, towards the upper part of the throat so that you can eject it from your mouth.